Hello everyone, welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall look at the general structure of a typical protein. Before we do that, Quirly would like to share another video of the crane couple that was preparing a nest for their baby or babies. Wow, look at the size of the nest. It's really increased here, right? Here's the nest at the beginning, and they were working and working and working, and now look at what a big piece of land they made for their nest. Very cool. Does look like 2 eggs, doesn't it? Here's I see one down here and one down here. Most likely 2 eggs. It'll be interesting to see how many babies are hatched. Well, that'll be interesting to see, Quirly. The video of a crane inspecting the egg, turning the egg. That's interesting. And then sitting on it to incubate the egg or eggs. Very cool. Let's have a look at the video. That'll be interesting. Very, very nice video. Thank you for sharing, Quirly. That was very interesting. Okay, so what is the general structure of a typical protein found in your body? So proteins are made of amino acids. So if this is an amino acid, it's joined to another amino acid and another amino acid and so on, giving you basically a string of amino acids, a macromolecule that we call a protein. Macromolecule. So there are 20 amino acids that make up a protein, and we shall talk about these amino acids, the building blocks for proteins, in another video. For now, it is enough to know that the amino acid, a typical amino acid, has an alpha carbon, and to the alpha carbon is attached the amino group or the amine group, and to the same alpha carbon there must be a carboxyl group, the acid. So the amino is the amine, the acid is the carboxylic acid. And then there is a hydrogen, that's your third group, and the fourth group attached to the alpha carbon can be any group that varies from amino acid to amino acid, and we call this the R group. Okay, so all the amino acids, you can see they have different R groups. Rest of the groups are the same, but the R groups vary from one amino acid to another amino acid. Okay, so that's what a typical amino acid is. Alpha carbon joined to 4 groups, your carboxyl, your amine, then the hydrogen, and finally some group called the R group. So how are these amino acids joined together to form a protein? You can see the amino acid has a carboxyl group, and that carboxyl group will react with an amine of another amino acid, and that will form a CONH, or what we call the amide. In organic chemistry, this is the amide group. But of course, in proteins, this would be called the peptide. This is your peptide group. And this the actual bond that connects the two amino acids is the one between the carbon of the carboxyl and the nitrogen of the amine. And this is called the peptide bond. So that's how amino acids are joined to one another by their peptide bonds. So in this case here, the carboxyl of glycine reacted with amine of aspartic acid to give you this dipeptide, a small protein. But in the second case here, it's the carboxyl of the aspartic acid reacting with amine of the glycine, giving you this dipeptide. So it's very important to understand that these two dipeptides, 1 and 2, are totally different in terms of structure as well as biological or even chemical properties. Okay, because their order is different. Here we have aspartic acid joined to glycine. Here we have glycine joined to aspartic acid. So you can see, just to make a dipeptide, just 2 amino acids joined together, the first position can be any of the 20 amino acids, and the second position can also be any of the 20 amino acids that are available to the cell. So that's 400 possible dipeptides, theoretical dipeptides. So you can imagine for tripeptides, there's 20 times 20 times 20, which is 20 raised to the power of 3. That's 8,000 possible tripeptides, theoretically possible tripeptides. The typical protein in the human body has about 400 amino acids. 400 amino acids. So how many proteins can we make out of that? That's 20 raised to the power of 400. And I don't even want to try to calculate that because that's a huge number. So it's a huge number of proteins that we can theoretically make by joining amino acids to one another. So once you get a dipeptide, then another amino acid with its amine group here can react with this. Here's the carboxyl part, and then you can get a tripeptide. And this reaction can continue and continue to give you a longer and longer protein. Although this is the reaction in terms of chemistry, we should remember in real life, in your cell, this carboxyl here of this amino acid is actually connected to a transfer RNA. And this carboxyl here is connected to another transfer RNA. So the reaction is really between this amine on one transfer RNA with this carboxyl connection to transfer RNA is actually through an ester. So it's really a reaction between amine and an ester. That's what happens in protein synthesis or translation. The amino acids are actually connected to transfer RNAs. So we shall talk about translation in a future video. Here's a protein with 1, 2, 3, 4, 5 amino acids. So let's look at some of the important definitions so that you can understand the structure of a protein and later on its function. So let's look at the structure of this simple pentapeptide, a protein with 5 amino acids. some important definitions. So here is the alpha carbon and then joined to another alpha carbon by a CONH, your peptide linkage. And then again, there's another alpha carbon joined to the previous alpha carbon by another peptide, and another alpha carbon joined by a peptide to the previous carbon, and finally the last or the fifth alpha carbon joined to the previous alpha carbon by a peptide. So here we have an amine connected to the alpha carbon that's not involved in peptide formation. So this is called the N-terminal of the protein. And here we have a carboxyl joined to the alpha carbon that's not involved in peptide bond formation. This is called the C-terminal of the protein. So the N-terminal and the alpha carbon and the amide and the alpha carbon and then the amide and then the alpha carbon and then the amide, all of these can be called the backbone of the protein. That's what we shall call it. This is the backbone of the protein. Alpha carbons joined to one another by peptide linkages. Okay, so this is the backbone of the protein. Now attached to the backbone are the R groups. The R groups are attached to all the alpha carbons. So here is an alpha carbon. Here is what we call the R group of the amino acid. And we'll talk about this later under amino acids. And here's the alpha carbon. Here is the R group of that amino acid, and so on. Here's the alpha carbon. Here's the R group. Here's the alpha carbon. Here is the R group. Here is the alpha carbon. Here is the R group. So basically a protein is a backbone. A backbone. To that backbone at the alpha carbons are joined the R groups. Okay, so we have the backbone, and sticking out of the backbone are the R groups. On one end you have the free amine, the other end you have the free carboxyl joined to the alpha carbon. So that's basically structure of a protein chain, and this structure is called the primary structure or sequence of a protein. This R group suggests that this amino acid here is alanine. So the first amino acid in this protein is alanine, amino acid number 1. Then the next R group, it belongs to cysteine. So amino acid number 2 is cysteine. Then the next R group belongs to lysine. So amino acid number 3 is lysine. Then the next R group belongs to tyrosine. So amino acid number 4 is tyrosine. And the last R group here belongs to histidine. So amino acid number 5, the last amino acid in this protein, is histidine. So we always number the amino acids in a protein starting from the N-terminal. We always start from the N-terminal. So here's number 1, 2, 3, 4, 5. And if this protein had, 150 amino acids, there'll be somewhere here 75, 86, 87, 88, and so on. Okay, so this is called the primary structure or the sequence of a protein. And this is the most important structure This is the most important structure. They will talk about other kinds of structures, but this is the most important structure for the polypeptide chain. And where does this information come from to tell the cell that this should be alanine, should be number 1, cysteine number 2, lysine number 3, and so on? This comes from the protein gene, the DNA. On your DNA, there is protein gene, and that protein gene, as we saw in last videos, is transcribed or copied into mRNA. So indirectly, this information came from mRNA, which came from the DNA. So let's quickly look at the formation of a protein in your cell, just the basic steps. And we'll look at the protein called hemoglobin. Hemoglobin. Hemoglobin has two types of polypeptide chains, the alpha chain and the beta chain. The information for the alpha chain is found on chromosome 16, and the information on making the beta chain is found on chromosome number 11. In a human adult, hemoglobin is made by the immature red blood cells that are in your bone marrow. So that's where hemoglobin is made, by the precursor red blood cells in your bone marrow. So let's follow the beta chain of hemoglobin. So here in your DNA, you have the template strand, one of the strands that is copied. This is in chromosome 11. So this strand is transcribed or copied, transcription. So the T becomes a becomes a U, C becomes a G, and so on. And that gives you the mRNA chain. And then if there are any introns, the introns are removed and the exons are joined together. And that's called RNA processing. And then, of course, there is caps and tails that are added to protect the two ends of the mRNA. And then the mRNA, as well as all the other RNA, tRNA, ribosomal RNA, and so on, gets transported out of the nucleus. This is your nucleus, into the cytoplasm. Once in the cytoplasm, on your ribosomes, and here's your mRNA, this information on mRNA is copied into amino acid. So AUG, for example, will, according to the genetic code, this is a codon for the amino acid methionine. So your first amino acid here would be methionine. GUU is the codon on your mRNA. According to the genetic code, it specifies valine. So this amino acid number 2 is valine. CAU specifies histidine. So amino acid number 3 is histidine. UUG specifies leucine. Amino acid number 4 is leucine. And so on and so on. The beta chain gets translated. This is translation, because we are converting nucleotide language into amino acid language. In a similar fashion, the alpha gene on chromosome 16 is copied into mRNA, and then that mRNA is translated into the alpha polypeptide chain. Alpha polypeptide chain of hemoglobin. So now we have the primary structure of the beta chain here. What is amino acid 1, 2, 3, and so on. The sequence, that's the primary structure. So if the protein is long enough, then it undergoes at the same time while it's being synthesized, it starts folding. That's called protein folding. So it may first form some simple structures such as beta sheets and alpha helices. This looks like a spring. This looks more like an accordion. These are called secondary structures. So the beta chain will make some secondary structures. The alpha chains will fold into some secondary structures. In case of hemoglobin, we only see alpha helices. We don't see beta sheets. So some proteins may have alpha helices and beta sheets. Other proteins may have both beta sheets and alpha helices. Other proteins may have beta sheets, no alpha helices. All kinds of combinations are possible. So these are called secondary structures. And secondary structures happen mainly because of hydrogen bonding on the backbone. Hydrogen bonding on the backbone. You see, the backbone is made up of CONH, and so you can see the backbone can hydrogen bond very nicely to give you your secondary structure. So hydrogen bonding is the most important force that causes the formation of secondary structures. And the protein continues to fold into more complex structures. This now forms what we call tertiary structures. So tertiary structure, in this case, we have not only hydrogen bonding, we have also other kinds of forces, the hydrophobic force that will force all the non-polar groups to go inside the protein and the polar groups on the surface. Then we have electrostatic, positive and negative attract, positive and positive repel. All those forces will also start coming together to give you what we call the tertiary structure of the beta polypeptide chain. In a similar fashion, you will get the tertiary structure of the alpha hemoglobin chain. So for many proteins, this is where the folding stops up till tertiary structure. You get a three-dimensional shape, the tertiary structure. However, for hemoglobin, there is another folding that happens, another assembly. This is really an assembly. The chains are already folded, but now two of these beta chains and two of these alpha chains will themselves come together to give you the hemoglobin molecule. Okay, the hemoglobin molecule. This is called the quaternary structure, of hemoglobin. And that's what hemoglobin is. It is made up of 2 alpha and 2 beta in case of adult hemoglobin. So the relationship between the alpha and beta chains with respect to one another, that's called the quaternary structure. So a lot of very important proteins that have very important functions in the body, they have quaternary structures. However, most other proteins have up till tertiary structures. Once a protein is made, then it will be transported to the right location in the cell. In case of hemoglobin, it's a cytoplasmic protein, so it stays in the cytoplasm where protein synthesis occurs. So that's basically what the three-dimensional or the shape structure of a protein is. The protein, string of protein, the polymer chain folds, and notice that one of the key things we see is because there's water everywhere around the protein. So all the polar groups, the ones that like water, are next to water on the surface of the protein, while the non-polar groups, the ones that don't like water, are buried inside the protein. Okay, so this is how basically a protein looks like in water. Now, a protein in membrane maybe is a bit different because in membrane, this is oil. There is no water here. So in this case, this part of the protein is non-polar, that is facing the oil, while the inside of the protein is polar, away from the oil. So just the opposite. Okay, so membrane proteins are different than non-membrane proteins, proteins that are found in your cytoplasm or nuclear mitochondrial matrix or nuclear plasm and so on. Very different from those in the membrane. So here are some of the key points you should remember when you talk about a non-membrane protein, which is the most common protein that we will talk about. So let's start with the N-terminal. At one end of the string of the polypeptide chain will be an N-terminal. That's your N-terminal. And then the string, you can see the alpha carbon, then the CONH, and then another alpha carbon. In this case, the R group is here. Here's the R group. Then again CONH, and then the alpha carbon, and so on. So this whole structure is folded to give you a tertiary structure. So at one end, there'll be an N-terminus, an alpha carbon to which is connected a amine. And then on another end, somewhere in the protein, there will be a C-terminus, a carboxyl joined to the alpha carbon of the last amino acid in the protein chain. And as we mentioned, all the groups that like water will be on the surface. Hydrophilic groups that like water will be on the surface. Hydrophobic or non-polar groups will be inside the protein, because that will give you the protein with the best, most stable structure. So as I mentioned, proteins may contain not just alpha helix. They could have alpha helix and beta pleated secondary structures, or they could have just beta pleated, or they could have just alpha helix secondary structures. So all kinds of combinations are possible. Now, in some proteins, especially proteins that are secreted out of the cell, secreted proteins such as antibodies, insulin, and so on, there is another possible structure, and that's called a disulfide bond. This is a covalent. This is covalent. This is very strong, very strong. So unlike hydrogen bonding or hydrophobic force or electrostatic, these are weak forces, weak. Disulfide is a very strong covalent bond, and that's formed between two cysteines, which we'll talk about under amino acids. And the reason why secreted proteins like antibodies have this is because these disulfides give the protein a stronger structure, give the protein a stronger three-dimensional structure, because these are very strong forces. So secreted proteins have disulfides because these proteins that are secreted out of the cell, they get into your bloodstream or other fluids. And there in those fluids, the environment might be very, very harsh, very harsh for the protein. It may unfold the protein, so that's why nature made sure that these secreted proteins are much stronger and they have disulfides. So that's basically the three-dimensional structure of a protein, some of the typical or general concepts. And these different proteins will have different 3D structures. Just like your knife has a certain structure, The fork has a certain structure, the spoon has a certain structure, and these structures will give you different functions. Okay, so the knife is really good for cutting, but it's not very good for eating your noodles or your spaghetti. While your spoon might be good for drinking soup, but you can't do that with your fork or your knife. Okay, so this is really very important. Shape of the protein or any biomolecule, macromolecule, determines the function of the protein. So proteins have a lot of functions for the cell. They carry out most of the functions of cells. And we'll talk about these proteins and their functions in future videos. So there are nucleic acid binding proteins that affect gene expression. That means they are going to affect the protein profile of the cell. And when the protein profile changes, the cellular activities are going to change. Enzymes are proteins that increase the rate or the speed of biochemical reactions. All of reactions that happen in your cell, most of these reactions are catalyzed by enzymes. Signal transduction proteins help the cells respond appropriately to its changing environment. Okay, very, very important role. Transport proteins carry out transports of all kinds of chemicals across cells and membranes. Defense proteins such as antibodies and complements protect us against microbes and other possible harm. Structural proteins provide support to cells and tissues. And then we have proteins that do not fall very well in these 6 categories. And these are the miscellaneous proteins, such as the protein ferritin that stores iron. It stores iron and then make sure the iron doesn't react to form free radicals. Okay, so this is basically an overview of a typical protein in your cell or in your body. Thank you for watching this video, and we hope you have a wonderful day. Hopefully we'll see you in our next video. Bye-bye.